Phoebe Gonzalez, November 7, 2017, Exercise 1, Dilutions and Viable Plate Counts. So here I have my work surface disinfected, wearing my safety equipment. Prior to that, I've washed my hands thoroughly. And I also have my equipment labeled and ready. So here, like I said, I have my tubes prepared. I had to improvise with my tube racks since the tube racks that was sent to me are not the correct size. I have my distilled water, alcohol, and my yeast or my Saccharomyces cerevisiae prepared. Sterilize my pipette with alcohol for 20 seconds. So I am going to be transferring the yeast solution to the tubes, but first adding distilled water to each tubes. So I am adding 2.25 milliliters of distilled water on each tube. I have my tubes labeled from 10.1 to 10.6. Next, I have my yeast solution, swirling it around to evenly disperse the organisms. Here I'm trying to organize my tubes, make sure the numbers are correct and in numerical order. Right, so from here I am transferring 0.25 milliliters of Saccharomyces cerevisiae into the 10-1 test tube. Swirling it around. And then from the 10-1 test tube, 0.25 ml of solution into the 10-2 test tube. And then from the 10-2 test tube, I am transferring 0.25 milliliters to the 10-3 test tube. And then from the 10-3 test tube, I am transferring 0.25 milliliters to the 10-4 test tube. And from the 10-4 test tube, I am transferring 0.25 milliliters to the 10-5 test tube. And lastly, from the 10-5 test tube, I am transferring 2.25 milliliters to 10-6 test tube. After transferring the Saccharomyces cerevisiae to this test tube, then I will be transferring the solution from the test tube to their to the agar plates, to the corresponding agar plates. Okay, so before transferring and using that pipette, making sure that the pipette is clean. So here, the first test tube 10-1, I am transferring 4 drops or 0.125 milliliters of Saccharomyces cerevisiae solution to the 10-1 agar plate. Then gently spreading the solution evenly over the surface of the agar. Next, I will be doing the same for the 10-2 test tube, transferring over to the agar 10-2 agar plate. And gently spreading the solution evenly over the surface. Next, we are doing the test tube 10-3, transferring to the 10-3 agar plate. And swirling it around to cover it evenly. Next, we are doing the 10-4 test tube, transferring 0.125 milliliters to the 10-4 agar plate. And spreading the solution evenly over the surface. Next, we have the 10-5 test tube, transferring over to 10-5 agar plate. And last but not the least, transferring 10-6 test tube to the 10-6 agar plate with 0.125 or 4 drops of the Saccharomyces cerevisiae. And swirling it gently to spread the solution evenly over the surface. Next, I will be setting aside the agar plates for incubation for at least 48 hours.